Hello, my name is Nadia Pazmiño. My topic is long-term effect of gene therapy of lower congenital amaurosis. Definition: mutation in RPE65 cause lower congenital amaurosis, a progressive retinal degenerative. This type is approved was device to replace the defective genes in event of monogenic disorder. Application in medicine. We measure visual acuity, contrast sensibility, color vision, and spectra. Sensibility of the investigation of visual level. Pros and cons. However, the durability of the benefit from RPE65 gene augmentation depends on protection against retinal regeneration. In one study involving human participants, intervention between the age of 11 or 30 years leads to protections against degeneration sustained sustain, improves in retinal function. Issues applies in equal expressions specific differences in the outcome of gene therapy or large unexplained, but the main reflex difference is pathophysiological mechanisms, vector tropics, or both. The rate of retinal degeneration in humans is varied, but relative or least spec is typical habitat in mouse and dog models. Study in animal has shown that improvement. In conclusion, gene therapy which right to an RPI65 vector improve retinal sensibility above the model. Thank you very much. Continue the project in 5 videos.
Eyes are the windows to your soul, and yet an estimated 500,000 children become blind each year. Genes, known as the blueprint of life, are a major determinant of eye diseases. The number one cause of inherited blindness in children is called Leber's congenital amaurosis, which affects 2 to 3 out of 100,000 babies. A clinical trial in the United States is looking to cure LCA with a type of gene therapy called CRISPR. LCA is a genetic disorder that affects vision at birth. If both the mother and the father pass on mutated genes, then the child will be born with LCA. These mutations target the photoreceptors at the back of the eye called the retina. As a result, light does not get converted into brain signals, causing severe vision impairment. The gene therapy CRISPR is programmed to target the mutated gene so it can be removed, replaced, or a new gene can be added. The clinical trial is testing the effectiveness of CRISPR on 18 subjects of different ages with LCA. The CRISPR treatment will be injected in the back of the eye to snip out the faulty gene. The hope is that the subject will repair the faulty gene in order to restore vision. Editas Medicine and Allergen is currently at phase 1 and 2 with the Brilliance clinical trial. The trial that started in fall of 2019 is said to last 3 years, giving 3 doses of the treatment to 18 subjects with LCA.
I think this therapeutic is very promising, as the only successful treatment of LCA has come from gene therapy. Even if CRISPR doesn't pan out for this disease, I think it's a step in the right direction, and the study of CRISPR in this context will help in future studies that involve it as well. People see headlines in the news claiming there can be a cure for congenital blindness. A lot of people get excited, especially when it involves gene therapy and CRISPR, which has garnered a lot of media attention lately. I'm hopeful though for a couple of reasons. First, there actually already exists an FDA-approved drug called Lexterna that is a gene-based therapy that can restore functional vision to a certain form of LCA. Second, the fact that the researchers believe in the drug enough to go into human trials shows there is some promise. But it is important to note that only 10% of drugs that go into human clinical trials eventually do become approved drugs, and this can take many years for this to occur. The treatment works by using a synthesized RNA sequence that targets a specific DNA sequence. The RNA is attached to an enzyme called Cas9, which cuts the DNA, which is then repaired by the cell. This treatment is done in vivo, or in other words, the human body.
Congenital amaurosis. It's a rare form of retinitis pigmentosa, which is a progressive blinding disease. Usually these babies are significantly impaired, and it's devastating because not only is their vision terrible at birth, but it gets progressively worse with age as the cells in the retina die off. A dog born blind with this very same condition. So the puppies that were born blind couldn't see their way around a room, and we tested the possibility of restoring vision in these little puppies by delivering the normal copy of the gene, which is defective. It's called RPE65. And lo and behold, these dogs began to see. And so those results led us to propose to test this same approach for treating blindness in young children. For these individual patients, it's so gratifying to see what they now can do that they could not do before. There are 2 things that I think are really going to be important from this. One, that will move forward with this particular disease and get approval for the drug that we've been developing. The other outcome that I think is really important is that this could be a stepping stone for developing a treatment for other blinding diseases.
Into an electrical signal, which is transmitted across the layers of the retina and then to the brain for visual processing. In an eye with achromatopsia, cone photoreceptors in the retina are unable to convert light into an electrical signal. The most common causes are mutations in one of 2 genes, called CNGB3 and CNGA3. In gene therapy, an AAV vector containing a normal CNGB3 or CNGA3 gene is designed to provide the instructions for normal CNGB3 or CNGA3 protein and restore cone photoreceptor function. By introducing a functional copy of a gene into a patient's cells, gene therapy is able to correct the underlying genetic defect that is causing disease. The functional gene, delivered by an AAV vector, is thought to remain stable for many years, which suggests that gene therapy has the potential.
In people with Leber congenital amaurosis, the RP under the retina, between the retinal pigment epithelium and choroid, where it enters the cells of the retina. The vector releases the normal RPE65 gene segment. The normal RPE65 protein is now manufactured by the cell. The RPE65 protein is released from the cell to vision receptors, restoring normal visual function.